In stage 2 of translation, we will now consider initiation. In initiation, we first begin with the 30S subunit. And here we're going to go over initiation in prokaryotes. And then in the end, we'll discuss the difference between prokaryotes and eukaryotes when it comes to initiation. So it begins with the 30S subunit. The 30S subunit has 3 sites. It has the E, the P, and the A site. The A site is the amino acid carrier site, P is the peptidyl site, and E is the exit site. So the first thing that happens is that we have initiation factors. We have initiation factor number 1. It's going to come and bind to the A site. And then we have initiation factor number 3 binding to the E site. We can remember where IF1 and IF3 bind by simply remembering that A can be denoted as the first site, P is the second site, E is the 3 site. So 3 will bind to 3, IF1 will bind to the first site. So when IF1 binds to the A site, it will prevent the binding of the tRNA. And when IF3 binds, it'll prevent the binding of the 50S subunit, or that larger subunit that's going to go right over here. Why do we want to prevent the binding of the 50S subunit and the tRNA? Because first, we want to effectively bind the mRNA. Because remember, the mRNA is coming from the DNA. So the DNA is transcribed into mRNA. mRNA is carrying that genetic information that's going to be translated into proteins. So we first need that mRNA to come and bind properly. Once it binds properly, then we can have the tRNA bind and the 50S subunit bind. But for now, we want to block that binding. So we use these initiation factors, and when they're bound, they block the binding of tRNA and the 50S subunit. So once these two are added in, they're added in position. So IF3 is at the E site, IF1 is at the A site, the mRNA is going to come in. The mRNA comes in and it binds with the help of the Shine-Dalgarno sequence or an initiation factor. In prokaryotes, there is a Shine-Dalgarno sequence that guides the mRNA. Whereas in eukaryotes, this is the difference. Eukaryotes simply will have initiation factors. So somewhere over here, we have the Shine-Dalgarno sequence. That sequence will help the mRNA align at the 30S subunit. So we can see we have this codon. It says AUG. What does AUG code for? Well, we know it codes for methionine. AUG is in line at the peptidyl site. The Shine-Dalgarno sequence is right over here. 5' end is here, 3' end is here. Why? Because the genetic code encrypted in mRNA is coded from the 5' end to the 3' end, so it must be read by the ribosome at the 5' end to the 3' end. And that first codon, that first code that we have is AUG. So now we have to bring in a tRNA. What's a tRNA? Quick review, what's a tRNA? A tRNA looks like this. It has this clover-like shape. This is just, oh, whoops. So it goes like this. We have this side, and it's like this, and this is 3', this is 5', and that 3' end is carrying an amino acid. This is the T arm, this is the D arm, and this arm is carrying that anticodon. So since we have AUG codes for methionine, we need a tRNA bringing in a methionine. So we see that over here. We have this tRNA. We see the anticodon, because if we have AUG as our codon on the mRNA, the anticodon will be UAC. And we see that right over here, the anticodon. And it codes for methionine, but we notice an F. Remember that right now we are talking about prokaryotes. Prokaryotes don't have methionine, they have N-formyl methionine. So we see this tRNA, it's carrying this methionine, and it's carrying it on the 3' end, and it's going to bring it in. But remember, we have this initiation factor 1 bound at that A site, which is inhibiting the binding of tRNA. So we have to get rid of this before the tRNA can bind. How do we do that? Well, we bring in another initiation factor, IF2. This IF2 is bound to GTP. When the IF2 comes and binds to that GTP, it facilitates it facilitates bringing in the tRNA as well. So IF2, when it comes in, it brings in tRNA with it. It's like bringing a friend to a party. The IF2 is going to bring the tRNA with it, and it brings it in. And the hydrolysis of GTP, so when we hydrolyze GTP, we go from GTP to GDP. This energy release is going to result in the hydrolysis of IF3 and IF2. They're going to be removed. They're going to be cleaved off. And we see that over here. They're cleaved off. IF2, IF1, IF3 cleaved off. We get GDP plus PI. Now, once that IF3 is gone, because remember, IF3 was bound to that E site, and it was inhibiting the 50S subunit from coming in. Now that it's gone, the 50S subunit can come in and it can bind. Now we're ready to go to synthesize the rest of this code and synthesize the protein. So let's look overall what happened. Initiation, second part of translation. First thing that we did, we prevented the tRNA from binding, and we prevented the 50S subunit from binding by using IF1 and IF3 on that 30S subunit. That 30S subunit has 3 locations, the A site, P site, E site. Next, mRNA comes in. The Stein-Delgarno sequence in prokaryotes, or the initiation factors in eukaryotes, allow for that binding of mRNA in the correct position from 5' to 3'. AUG is in line with the peptidyl site. Now we're going to, since this is the codon, we must find the anticodon, which is going to be UAC. So our tRNA is going to have an anticodon of UAC, and AUG stands for methionine. It codes for methionine. So at the 3' end, we have to be carrying a methionine, and we see that. But since we're talking about prokaryotes, we have N-formyl methionine. This is the anticodon, tRNA, carrying N-formyl methionine. But since we have this IF1 and tRNA can't bind if IF1 is there, we bring in IF2. And IF2 brings in the tRNA with it. And this hydrolysis of GTP to GDP allows for the removal of initiation factor 1, 2, and 3. And the 50S subunit comes in. And we get this final result where both the 50S and the 30S ribosomal subunits are bound together. Our mRNA goes from 5' to 3', and it's at that peptidyl site with AUG. We have our first tRNA in place with that N-formyl methionine, and now that's the end of initiation. Now we're going to go into elongation, which is another process. What's important to note, most importantly, is that we're utilizing a GTP. So we're utilizing 1 GTP in order to initiate, in order to start the process. But remember, the tRNAs that are carrying that amino acid have been so we create that amino acidyl tRNA, and charging a tRNA with amino groups utilizes 2 ATP equivalents. So far we have used 3 ATP equivalents, and we still have 2 more stages to go.